All right. So yesterday I got started on the topic, a really cool topic about replication of DNA in eukaryotic cells. And I'll remind you that the chromosomes in eukaryotic cells are linear. And at the ends of those chromosomes, we have special structures called telomeres that are simply thousands and thousands of repeats of the same short sequence of DNA over and over. And those telomeres are made by a special enzyme called a telomerase. And the telomerase works by carrying its own template. So when I say template, what a template is something that's being copied. It's something that's being copied. Okay? Now I'm going to show you a figure which I hope will not scare or confuse you. But carrying its own template is important. Okay? Because at the very end of the DNA, there's no sequence, and you want to put sequence out there. So you've got to copy something. So the telomerase carries something to be copied, and it copies it, and then it slides down, and it copies it again, and it slides down, and it copies it again. And that goes on thousands of times. And the template that it copies is not DNA. The template that it's copying is RNA. Now, this is an example of an enzyme that is copying RNA and making DNA from it. We haven't talked about this enzyme before, but an enzyme that copies RNA, that means it's reading an RNA strand and making a complementary DNA to that strand. When we talked about the central dogma the other day, you saw something called reverse transcription, which was the backwards way of making RNA. It meant that you were starting with RNA and making DNA, and I said that retroviruses like HIV did that. Those viruses that do that have an enzyme called reverse transcriptase. That's a name you should know. Reverse transcriptase. And a reverse transcriptase is an enzyme that copies RNA and makes DNA from it. Now, as you've started to see from what I've told you here, telomerase is a type of reverse transcriptase because it's copying an RNA, and the RNA it's copying is the thing that it's carrying in its back pocket. Telomerase is a reverse transcriptase because it's copying RNA to make DNA. Now, as I noted yesterday, telomerase is active in embryonic cells. It's active in stem cells. And unfortunately, it's active in cancer cells. So we don't want to just randomly go and make telomerase active, because if we do, we may cause bigger problems than we're going to solve. And as I briefly mentioned at the very end yesterday, one of the strategies for knocking out some types of cancer is to actually inhibit telomerase, because that will let a cancer basically burn itself out because it's not making the telomeres. If you think about it, a tumor cell has to keep making telomeres, because if it doesn't make telomeres, what's going to happen? They're going to shorten and the cell's going to die. So if we inhibit telomerase in a cancer cell, we might be able to have the cancer cell basically kill itself. And in some types of cancer, this strategy looks like it actually might work. Okay? Questions about that? Yes?
Why would it kill a cancer cell, not a normal cell?
A normal cell doesn't have telomerase. So if you think about it, a cancer cell is dividing very rapidly. A normal cell is not dividing so rapidly, and the normal cell doesn't have that as an issue. Okay? It's a thought. And there are some strategies in some places where it looks like that might actually work. Okay? Other questions? Okay. Well, there's something else about DNA replication I want to tell you that's pretty cool as well. Okay? And this involves another protein that is found in eukaryotic cells but not found in prokaryotic cells. Okay? Now, I've skipped over this part about DNA repair, and I'm going to come back and talk about DNA repair in a minute. But before I talk about DNA repair, I want to talk about this protein because it's relevant to DNA repair. Let's imagine I'm a eukaryotic cell. I'm getting ready for the big dance, meaning I'm getting ready to divide. In order for me to divide, I've got to replicate my DNA. And I go, and there's actually a cycle. It's called a cell cycle that our cells go through that controls the different phases that the cell goes through. And one of those phases involves replication of the DNA. And replication of the DNA, as you might imagine, is the most important thing that has to happen during that cycle because, first of all, if replication doesn't happen, the cell can't divide. Second of all, if the cell has a problem during the replication process, then the DNA may be damaged, and damaged DNA can be real problematic. Okay? We'll talk later about things called oncogenes. And oncogenes are genes that are mutated, and as a result of the mutation, cause cancer. So you can imagine then if we have problems that are occurring during the DNA replication cycle of a cell, that we might be mutating some important genes, and if we mutate those important genes, we might be causing that cell to become cancerous. Well, fortunately, our cells have built into them what I call a quality control system. Yesterday on the song that we sang at the very end, you saw something called P53 being the QC system for the cell. QC stands for quality control. Okay? Quality control. So now I'm going to tell you about this very important protein called P53 and its role in making sure that replication is happening properly. It's really cool what it does. Okay? I told you there's a cell cycle. It goes through several phases, one of which is replication. When the cell goes through the replication phase, which is called the mitotic phase, the synthesis phase, not the mitotic phase, the synthesis phase. When the cell goes through the synthesis phase, where the DNA is being made, at the very end of that phase, there's a quality control moment. And the quality control moment, the cell is able to ask, at the molecular level, has replication proceeded properly? Has replication proceeded properly? And there's actually ways that cells can read the signal and decide if the DNA has been completely replicated properly. If the signal is that the replication has completed and it's completed properly, then the cell will proceed to the next phase, which will involve division, replication. On the other hand, if the cell gets a signal that replication has not completed properly, it's stuck because there's a problem with the DNA, or the polymerase can't resolve this chemical that's been added to the DNA and it can't fix that problem, then this protein called P53 steps in. P53 is the quality control protein for DNA replication. If it gets the signal, everything's okay, P53 says, go ahead, divide. If P53 gets the signal that there's a problem, P53 will stop the cell cycle in its tracks. It will not let the cell proceed to the next phase, because if it did, the cell might likely become cancerous. P53 will then stimulate the production of DNA repair proteins. We're going to talk about those in just a minute. It will stimulate the synthesis. The cell has the ability to synthesize proteins to repair DNA. Those proteins will be made when P53 says, wait, we've got a problem. Everybody with me? At that point, the cell has two possible things that can happen. The repair proteins will go and try to repair the damage. Either they will repair it and report back to P53, everything's okay, and the P53 says, okay, go ahead and divide. Or they will be unable to repair it, and they'll report back to P53, we can't fix this mess. In that case, P53 will stimulate the cell to commit suicide. Okay? P53 can cause the cell to commit suicide. This only happens in eukaryotic cells. And it's very important for multicellular organisms. Because if you think about it, one cell in a multicellular organism that decides it's cancerous causes a problem for the entire organism. P53's role is very, very important. So P53 says divide or hold on. And if it says hold on, then it either gets damage repaired, in which case it says divide, or damage doesn't get repaired, and it says commit suicide. Suicide, cellular suicide, is called apoptosis. A-P-O-P, the second P is silent, A-P-O-P-T-O-S-I-S. The second P is silent, just like the P in pneumonia. Okay? Now, that is a very important role for P53. You might ask the question, what happens if something happens to P53? What if P53 itself is damaged so that it can't perform this function? What do you suppose would happen in a person who had a cell that had a damaged P53? That cell would be much more prone to having cancer. Because P53 is stopping a lot of processes from happening that would result in cancer. And that's exactly what happens. In fact, when we look at many tumors, what we see in many tumors is that they actually have a damaged P53. Unable to do quality control, that cell went forward when it shouldn't, probably caused a tumor. Yes, question? Okay. So what's the difference between a cancer cell and a tumor that's benign? A tumor that's benign doesn't divide uncontrollably and doesn't spread and cause problems. So something that is able to do that can metastasize, and that's where cancer kills you. So a benign tumor can't do that. Okay. So, pretty cool stuff, right? We'll see that there's, and by the way, P53 is an example of an oncogene. You mutate. Remember I said certain genes that you can mutate, and if you mutate them, you'll be more prone to have cancer? P53 is it's actually what we call a proto-oncogene. I'll explain that distinction for you later. But if you mutate P53, you can get an oncogene. Okay. All right. Now, don't need to worry about that. And the last thing I want to talk about is DNA repair, and I'll say a very brief word about recombination. Okay? So as you can imagine, DNA repair is pretty important because it's one of the things that bails out the cell in the scenario I just gave you, right? P53 says there's damage. P53 tells the cell, make repair proteins, and the repair proteins come and try to fix the damage. So DNA repair proteins are very important. Now, I told you that P53 is only found in eukaryotic cells, but the repair proteins are found in all cells. So eukaryotic cells use P53 as one way of turning them on, but all cells have DNA repair proteins. The ones I'm going to show you are found in E. coli, a bacterium. Okay? Before I talk about that, let's talk about how you can damage your DNA. I went out to dinner on Sunday night with a former student from this class, and she's working up in Portland right now, and she told me there was one thing in this class that stayed with her. She's been out of the class for 4 years, ever since the class. And it's what I'm getting ready to tell you right here. Okay? This has stayed with her. Okay? What you see on the screen is what happens when you go to a tanning booth. Now, I'm not going to ask for a show of hands to see who goes to tanning booths. I know entirely too many people do that. Okay? Tanning booths are designed to give you a quick tan, and tanning booths do so by basically frying your cells. They're inducing your cells to make a pigment, okay? But in the process of doing so, they damage your DNA. Ultraviolet light is damaging to DNA. You might say, does sunlight damage DNA? And the answer is yes. So you want moderation. And yes, you do want some sunlight because sunlight helps you to make vitamin D. Sunburning, I don't recommend. Tanning boothing, I don't recommend at all. Tanning boothing tends to use a stronger dose of ultraviolet light than what you get from the sun. That's how it works so fast. Okay? When you hit DNA with ultraviolet light, here's what happens. If you have two thymines that are next to each other on the same strand, when ultraviolet light hits them, they can react as you see right here on the screen. This is a thymine, this is a thymine, and this is a thymine dimer, meaning that there's now a covalent bond between that carbon, that carbon, that carbon, and that carbon, and this guy is something that looks really odd to a DNA polymerase. It looks really odd to a DNA polymerase. DNA polymerase won't be able to read that properly when it goes to copy it. If that dimer is not fixed, you're very, very likely to have a mutation at that point. Well, what if that dimer is in your P53, folks? Do you see problems in paradise? Yes. It's only one dimer. It's only one gene. What's the chances? How many cells do you have on the surface of your skin? Okay? Yeah. All right. So, thymine dimers are problems. I'm showing you the problems. I'm going to show you some of the solutions. But the solutions don't mean that you go to a tanning booth. The solutions mean that if you don't overload them, they probably will work for you. And if you overload them, like anything else, you're probably going to have some problems. That's why the tanning booth part is the problem. Okay. Well, how do we fix damage in DNA? This is the job of working with repair proteins. Okay? Here's one type of damage that might occur. This is a type of damage that is not a thymine dimer. I'll talk about thymine dimers in a minute, but thymine dimers illustrate the importance of taking care of your DNA. Taking care of your DNA means no tanning booths. It means don't smoke. Because just like you saw, thymine dimers can happen. If you smoke, you can put chemicals attached to your bases, which are even bigger problems. Okay? Well, here's a problem that can arise as a result of a completely innocent thing. This can happen if DNA polymerase, for example, goes along, does its thing, okay, and it misses something in the proofreading. So the proofreading is really good. I said that what we have, let's say 1 in 10 million to 1 in 100 million, that's the error rate for a DNA polymerase. Keep in mind that you have 7 billion bases or base pairs of DNA. You're making some of these all the time. So repair becomes very important to fix these. If you have unrepaired damage that's happening inside of your DNA, it's important that it get recognized first, removed second, and replaced third. Okay? Now there's a bacterial system, it's not actually shown on here, but there's a bacterial system that's called the Mut LHS system. Mut L, H, and S, those 3 letters. And those 3 letters correspond to 3 different proteins. Mut L, actually I won't even distinguish the proteins. They do the following functions. Recognize the error, meaning there's a bulge in the DNA because I've got a G paired with a T. Like we talked about yesterday, that's not going to base pair properly, so a bulge is going to exist in the DNA. And it's escaped the proofreading, so that means that T is now attached to something else, and now it's in the middle of a strand of DNA. Okay? The system will, first of all, recognize that bulge. Second of all, it will make a cut in the strand that has the problem. It will remove that piece, and then the polymerase will come along and fill in the gap. That's what the Mut LHS system will do. That one's important because, again, polymerase is really good, but it's not perfect. And because it's not perfect, we want to try to fix errors that it's making during its replication process. The system I just described to you is in E. coli, but we have a system in our cells that's almost identical to it. They have slightly different names, but we won't worry about that. Okay. What about thymine dimers? Well, thymine dimers, as I said, are kind of odd structures. And they're depicted on this screen here as having a bulge in the DNA. Most mutations and most damage to DNA will result in an unusual structure in the DNA. And that's what you're seeing, an unusual structure in the DNA. The unusual structure is a target for a DNA repair system. Here's a repair system called ABC. Very simple to remember. Okay? ABC. The ABC system recognizes the problem. And in this case, the problem is quite clear because there it is right there. You'll notice it makes cuts on either side of the problem. And then it removes that. Once it removes that, look how easy it is for it to fill in the replacement. As long as I don't overwhelm this system, I will repair the damage that occurs to my DNA that are thymine dimers, as long as I don't overload that system. And if I overload that system, there's going to be some of these that are going to escape and are going to cause mutation because the polymerase will come along here and say, oh, I think that's a G, or whatever. It's not going to accurately read what's there, and it's going to make a mistake. And that's going to be a problem. Okay? Okay. So that system that I just described to you is called nucleotide excision repair. The ABC system is nucleotide excision repair. There's other damage that can happen to our DNA. These are all horror stories. Other damage that can happen to our DNA. Let's say you are a smoker. One of the things that smokers get an abundance of that they really don't want is something called reactive oxygen species, also called an ROS, reactive oxygen species. Reactive oxygen species are about as bad of a thing as you can have for a cell. Okay? Because a reactive oxygen species is basically an oxygen molecule that is O2 that has an extra electron. And things that have an extra electron, extra unpaired electron, are extraordinarily reactive. They will react with the first thing that they bump into. Reactive oxygen species. Everybody knows about Linus Pauling and vitamin C, right? Linus Pauling says vitamin C is a wonder drug because you take a lot of vitamin C, he says you can cure almost anything. Do you know why Linus Pauling felt that way? Because what? He didn't what? He didn't know He didn't know he peed it all out. That's partly true, okay? But that's another story. You do pee out most of the vitamin C that you take. He was trying to get the body's dose of vitamin C up. And the reason he was trying to do that was because vitamin C is an antioxidant. It protects against oxidative damage. Now, when you get oxidative damage, there's a variety of things that can happen in the cell, but the worst thing that can happen in the cell is you oxidize your DNA. Linus was interested in antioxidants a lot. Vitamin C is one of them. Vitamin E is another one. They both protect against reactive oxygen species. I'm going to give you an example. If you look at DNA, DNA has A, G, C, and T, right? Reactive oxygen species really seem to like guanine. They really seem to like guanine. They will react with guanine and create something called 8-oxoguanine. That's an important molecule. So they convert guanine from just plain old guanine within the DNA into 8-oxoguanine in the DNA. Who cares about that? Well, you should care about that because when the cell goes and it copies, that is the polymerase copies 8-oxoguanine, it thinks it's a T. And it puts an A across from it. You just made a mutation. A smoker who's getting a lot of reactive oxygen species is making a lot of 8-oxoguanine. The more 8-oxoguanine you have, the more mutation that you have. And the more mutation you have, the more likely you are to create a cancerous cell, just as we've been going through and saying. Okay? One of the ways of fixing such damage is by actually removing the damaged base. This is called base excision repair. And it simply involves an enzyme clipping out the damaged base and then doing a repair system, which is basically a polymerase that fills it in to fix it. Now, I'm not expecting you're just going to memorize these repair systems. That's not my point of showing them to you. I'm showing them to you so that you understand the importance of these repair systems and you understand the importance of damage to DNA. Damage to DNA is a giant problem, and damage to DNA is essentially the problem with cancer. So when you see people who are worried about breathing clean air, or they're worried about having clean water, or they're worried about the stuff that's in their food, the ultimate reason that they're worried about those is because each of those can cause damage to the DNA of your cells. Okay? More damage, more likely you'll have cancer. Okay. Let's see. And I think I was going to talk about recombination, but I think I'll pass on that. Okay, so let's not talk about recombination. Any questions before I move on to the next thing? Yes? Okay. What's the difference between the first system that I talked about, where the polymerase has missed something, and the last one, I think you said, right? Okay. Okay. Yeah. Yeah, good question. In the case of the second one, so nucleotide excision repair is what's happening in the first one as well. Okay? So you can classify them together, all right? The mechanism is slightly different, and the proteins involved are slightly different, but in both cases, you're removing a piece of a strand and then replacing it. Okay? Okay. Let's turn our attention to the synthesis of RNA. Oh boy, right? Can't wait. First of all, notice the title at the top of this. RNA synthesis is transcription, not translation. RNA synthesis requires ribonucleotides, not deoxyribonucleotides. Okay? DNA synthesis was catalyzed by a DNA polymerase it's not too much of a stretch of your imagination to say that RNA synthesis is catalyzed by an RNA polymerase. RNA polymerases turn out to be a little easier to understand than DNA polymerases, partly because they can start synthesis all by themselves. DNA polymerases couldn't do that. RNA polymerases can. Features of RNA synthesis, okay? The process is called transcription. The enzyme, you can leave out that DNA dependent. The enzyme is RNA polymerase. Okay. E. coli cells are really simple. They have one RNA polymerase. Eukaryotic cells are a little bit more complicated. They have 3 RNA polymerases, and there are hints of others. Okay. All the different types of RNA in a bacterial cell are made by the same RNA polymerase. In our cells, we have specialized polymerases for specialized RNAs. We have one that makes tRNAs, for example. Okay? The nucleotides are ATP, GTP, CTP, and UTP. Those are ribonucleotides. Primer is not needed. But a DNA has to be there because it has to copy something, right? It's copying a DNA. I said that replication of DNA starts at a special sequence. What did I call that sequence? An origin. Okay? Extra credit there. Starts at an origin. RNA synthesis starts very close to a special sequence we call a promoter. Very close to a special sequence called a promoter. The aim of DNA replication is to replicate the entire DNA molecule. The aim of transcription is to make either one gene or a small set of genes, not the entire chromosome. Okay? Only a part of the DNA is copied in transcription. Okay? All right. Let's talk about promoters to start. Now, most of what I'm going to tell you about transcription is actually what prokaryotes do. And the reason for doing that is because, first of all, it's the one that we know the most about. But second, transcription in our cells, that is eukaryotic cells, is enormously more complicated than it is in prokaryotic cells. I'll say a little bit about our cells, but most of what I'm going to tell you is what happens in prokaryotic cells. All right? In prokaryotic, in both cells, we have sequences called promoters near where transcription starts. Okay? But I'm going to talk about prokaryotes first. Let's talk about promoters. Here's a promoter sequence that's found in prokaryotic cells. Here's a promoter sequence. All right? What is this? Well, if we think about transcription, transcription has to start somewhere, right? So what you see on here are several different genes in E. coli. Their names are on the left. No, you don't need to memorize those. And over here where it says transcription start site, that's the first nucleotide, okay, that's copied and put into the RNA that's being made. In each case here, we're talking about messenger RNAs. Okay? So the first nucleotide here, they're all shown in yellow. Now, interestingly, if we're looking now at the DNA sequence in that region, if we look about 10 nucleotides ahead of that region, called the -10 region, and that's in the 5' direction moving leftwards, we see a common sequence that's not identical in each case, but it's similar. Okay? It's similar. That sequence, if you look at it, and you say you compare the sequence from this one to this one to this one to this one, and you start adding up what the most common bases are that are in that sequence, we see that the first base in that sequence is a T 79% of the time. We see that the second base in that sequence is an A 95% of the time, and you can see all the other distributions there. The fact that this sequence appears over and over and over at about the same place where the transcription starts says that this sequence must be important. This sequence is part of the promoter. Okay? The promoter is something that in a prokaryotic cell is where the RNA polymerase is binding. It's grabbing a hold of the DNA at this point. And in order for the RNA polymerase to copy DNA, by the way, when RNA polymerase copies DNA, it only copies one strand. It doesn't have to do the replication fork. It's just copying one strand. To copy one strand, what's it got to do? Well, it's got to pull the DNA strands apart. Because it's only going to copy, let's say, the bottom one here. The top one is just going to pull out of the way. Well, if you've got to pull something apart, wouldn't you like to find the easiest way to pull it apart? Cells are lazy just like we are. What's the easiest base pairs to pull apart, folks? ATs only have 2 hydrogen bonds. GCs have 3, right? Look at that sequence. Do you see any ATs, GCs that are commonly appearing in there? You see a scattering occasional one, but for the most part, you don't see Gs and Cs there. It's easier to pull these guys apart. The -10 sequence in bacterial cells is called the Pripno box. And you'll notice it's very AT-rich. Not coincidentally, that's the easiest sequences to pull apart. You might say, well, why aren't all of the sequences identical? Why are some of them have a TAC, TGT, and others have TAT, TAT? Why aren't they identical? What you're starting, if you think about that question, what you're starting to ask is a very important question, and that is, why doesn't the cell make everything at the same level? Do you suppose that the cell would need as many DNA polymerases as it would ribosomes, things that make proteins? The cell has to make a lot of protein. The cell is going to have a lot of those different needs to make proteins. It's going to have a lot of ribosomes. It only needs a few DNA polymerases. So if it makes as many DNA polymerases as it makes ribosomes, it's wasting energy because most of those DNA polymerases aren't going to be needed, right? The cell doesn't need to make as many of a certain protein. It doesn't need to transcribe as many messenger RNAs of that protein. Now, what happens here, the closer one of these sequences is to this ideal sequence, the more commonly RNA polymerase will copy it. We call that a strong promoter. A strong promoter is something that results in the production of a lot of messenger RNA. And the more Gs and Cs we see in that sequence, the weaker that promoter is, meaning the less messenger RNA that will be made. And that makes sense for something that the cell doesn't need a lot of. Control over the amount of protein, or I'm sorry, the amount of messenger RNA that's being made is something we call control of gene expression. Control of gene expression. And we'll see several ways in which this occurs. You're seeing the first of those right now, which is that certain genes have different promoters. Some have strong promoters, some have weak promoters, and those are a reflection of how much of that protein the cell ultimately needs. Okay, a lot of material for today. You guys ready for the song for the day? Questions? This is a fun one today, and I can't sing it worth a crap, but it never stopped me from trying. You guys know the Village People? YMCA? You got it. Okay. Phosphates are in nucleotides. I say phosphates cover bases inside. I say phosphates span the 5 and 3 primes. There's no need to be real mixed up bases. Carry info, you see. I say bases are all complementary. I say bases like A, T, G, and C. They have got to be all paired up. Boom, 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 boom. It's fun to play with some bDNA. It's got a boatload of GCTA. It's got everything a polymerase needs when you melt all the A's and T's. It's fun to play with some bDNA. It's got a boatload of GCTA. You can make RNAs with a polymerase just by pairing up U's with A's. Proteins full of amino A's. I say proteins come from mRNAs. I say proteins require tRNAs. There is more you need to translate codons. Like our friend UAC, I say codons come in clusters of 3. I say codons have one base wobbly. Now you can go forth and translate. Boom, 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 boom. It's fun to play with some bDNA. It's got a boatload of GCTA. With those, editorial comment there, with those hydrogen Bs and right-hand helices, anti-parallel 5s and 3s. I think that's enough. My voice is giving out.